So, what are the mechanisms behind PACVS? And sort of the leading hypothesis among people in what you'd call the medical freedom movement, this would be what could be termed the spike hypothesis, that the symptoms were a result of persistent spike protein. That view has There are some issues with that view. In recent months, there's been studies that have shown that one spike protein load in the context of long COVID does not have any association with their symptom profile. So the severity of symptoms. However, that is measured in the bloodstream, so it's possible there's persistent spike elsewhere. Um, the spike hypothesis, it does somewhat align with viral persistence, which is a leading hypothesis for the mechanism of long COVID. In the case of viral persistence, you have actual latent SARS-CoV-2 in your cells that your body hasn't quite cleared. And the spike hypothesis suggests that there is a reservoir of spike or its genetic material, which is leading to its persistence. And I'm not too sure about the spike hypothesis myself. I thought it was very convincing a little while ago, and you could look at the mechanisms that were shown in lab experiments for the spike protein, such as interference with estrogen receptor alpha, as well as some of the neurological symptoms. And you could generally map what we saw at a laboratory experimental level to symptoms. So that, to me, it looked quite convincing, and that still might factor into the disease mechanism. However, I'm more interested in the metabolic component because I think that's more directly measurable. It has been very difficult to measure spike, even in the studies that you do see persistence, and we did a recent meta-analysis on this. It's at very low concentrations. That doesn't mean it doesn't have an effect. It could still have an effect even at these low concentrations. However, the therapeutics which have developed around the spike, such as immunoprecipitation, there's some use of monoclonal antibodies. I'm not quite sure how those trials are going, but immunoprecipitation, it seems to have not performed at the level it was expected to. So that's one possible mark against the spike hypothesis, that people were trying to immunoprecipitate these things out, and it didn't seem to do much. However, it's still possible. I think the metabolic focus makes more sense. It's directly observable. The experiments, they're not quite point of care or common. Some of it requires complex metabolomics, but you can look at proxies such as lactate threshold, and there's even tests you can do using apps that give a good proxy for your lactate threshold or VO2 max, which I think we will apply in a decentralized clinical trial manner. So, for the mechanisms that are relevant to metabolism, you do have impaired oxygen delivery. That is due to the coagulopathies and endotheliopathies, which is possible it traces to the spike protein. An additional consideration for some of these studies showing either no spike protein or no relationship between spike protein level and symptoms is that if spike protein gets incorporated into a fibrin resistant microclot, then it might not show up on the assays that they use, which are typically immunofluorescence. They have an antibody which sticks to the spike, and that um, has a fluorescent protein at the other end, so you're able to see the signal. Those the formation of clots might interfere with that. However, there's been on the actual coagulopathies, there's been limited clinical validation of that. There have been some studies which have shown that people have gotten better with this triple anticoagulant therapy. Therapeutic apheresis, that is basically switching out your blood or sometimes cleaning out your blood. This has largely been null in the literature. So that was a surprising finding legitimately. So we're not sure. I think it is one contributing factor out of many. When you look at these issues such as ME/CFS, long COVID, these are fundamentally diseases. I like to think of chronic fatigue. There's different ways that energy production can go wrong. ME/CFS is a class of different disorders which affect your ability to produce ATP. So, that's sort of a very simple elementary school view of it, but I think it holds up. And so with long COVID and with PACVS, in addition to the issues in oxygen delivery owing to the cardiovascular system, there are also alterations in mitochondria. This could be due to the spike protein itself. We don't know. However, you do see that people's ability to metabolize fat, it drops considerably. And as you know, fat is a very rich energy source, 32 ATP per palmitate molecule. And so if you lose that capability, you lose a lot of energy production. So that is one contributor. Also, the lactate threshold. Basically, this goes through the floor with PACVS and long COVID. So possibly interventions which target this, and there are a few, such as buffering with baking soda, or cordyceps, or some intentional breathing exercises, like buteyko breathing, though that's controversial. These might be able to raise VO2 max. In the case of breathing, raise your end-tidal CO2, so you're actually able to get more oxygen into the cells. It's like the CO2 and the oxygen is like potentials of a battery. You have to have a difference between positive and negative in order for there to be a current. So that's how I see oxygen and CO2 in the body. And the issue with you see a lot in long COVID is their end-tidal CO2 is quite low. So the CO2 is the signal for the oxygen to dissociate from the hemoglobin molecule and enter the cell. If that signal is not as strong, you get less oxygen entering the cell. And so that's a potential way in which the ATP production gets compromised, because aerobic respiration is far more efficient than anaerobic respiration. And yeah, anaerobic also carries with it the production of acid, lactic acid, and that causes the burning sensation. So this is what a lot of people with long COVID and PACVS experience. So that is my brief recap of some of the mechanisms of PACVS, particularly focused on the metabolic part. which I think is the most interesting and clinically the most promising. There's some quite good research. We have summarized it in a protocol that is the vital scan protocol. I will get to that in another video.